Hello everyone, I am Snigdha Bharadwaj, Assistant Professor, ITS College of Pharmacy. So, I welcome you all, and in this session we will be continuing with the physical degradation and chemical degradation of the drug products and formulations. So, let us get started. So, basically in our earlier session we have discussed about various type of physical degradation and the factors affecting physical degradation and stability. In this particular session, we will be focusing on chemical degradation and the factors. So, it is the chemical degradation is the separation of chemical compounds into elements or simpler compounds. Change in the chemical nature of drug is called chemical degradation. When the drug is chemically changed during the storage, when we formulate the preparation and we keep it for further use, and that period is called the storage period. So, sometimes chemicals may get degraded during the storage time. So, it is not mandatory that we can see any chemical changes or reaction in terms of color change and odor change. So, sometimes the chemical incompatibilities may be seen during the storage period between the drug and the component. So, basic pre-formulation studies must be employed for identifying these interaction, possible incompatibilities between the API and the excipients. So that the stability will be more for the formulations. So, type of chemical degradation are like what type of reaction may occur during the storage in the product. Hydrolysis, oxidation, carboxylation, decarboxylation, isomerization, polymerization. So, we will discuss these chemical reaction one by one. So, hydrolysis. First, the drug with ester and amide group react with one molecule of water and undergoes hydrolysis. So, basically hydrolysis means reaction with the water molecule. A few drug with undergoes hydrolytic pathway are given below, like esters, in esters, aspirin, procaine, atropine. These are few examples. Amides, chloramphenicol, ampicillin, cephalosporins. So, these are basically the drugs or API which are prone to hydrolysis during the storage or during preparation or any kind of. So, ideally we generally take the preventive measures to avoid these type of instabilities. So, protection against hydrolysis can be done by adding buffers, complexation, removal of water. So, these are the approaches we generally opt to reduce the hydrolysis reactions in the preparations. So, ester hydrolysis, this is the common equation. RCOOR ester plus H2O will make RCOOH and ROH, acid and alcohol, right. So, example of drug aspirin, atropine and procaine. Amide hydrolysis will give you this is the amide, and in presence of water it will make the amine and the acid. So, these are the example chloramphenicol and barbiturates. So, generally the reaction form, the reaction takes place like this. So, types of hydrolysis are of two types, ionic hydrolysis and molecular hydrolysis. So, hydrolysis occurs, ionic hydrolysis occurs when the salt of the weak acid and bases interact with water to give either alkaline or acidic solution. So, basically ionic hydrolysis refers when the salt of the weak acid in the reaction and bases interact with water. They are interacting with water molecule to give either alkaline or acidic solution. For example, CH3COOK gives alkaline, while codeine phosphate gives acidic solution when interact with water. So, whenever they interact with water, they will give in either alkaline or acidic type of solutions, which considered to be degraded form of the formulation. So, molecular hydrolysis, it is a much slower and irreversible process. It is catalyzed by hydrogen or hydroxyl ion, example the local anesthetics, amethicone and benzocaine. So, these are the drugs which follows, and sometimes these particular cases we observe molecular type of hydrolysis taking place. After this hydrolysis, oxidation. So, addition of oxygen or removal of hydrogen from the drug molecule is known as oxidation. So, either addition or removal of addition of oxygen or removal of hydrogen are termed as oxidation reaction. So, oxidation involves the removal of the electrons from the molecule, right. So, drugs that decomposed by the oxidation pathway are ascorbic acid, clove oil, vitamin A, vitamin B12, morphine and many more. So, these are few examples which follows this oxidation reaction for and become degraded. So, oxidation is prevented by using antioxidants or chelating agent. So, we generally add chelating agents and antioxidants to the preparation to avoid these types of reactions. Antioxidants either get itself oxidized or prevent the overall oxidation environment. Oxidation reaction are known to occur in presence of oxygen, trace metals, hydrogen ion and OH ions. Preventive measures should aim at eliminating the influence of these factors on the drug. So, our target is to avoid these particular type of factors which affects the oxidation reaction in a particular drug, and it may cause the drug degradation. So, we generally add antioxidants or chelating agents to overcome this oxidation problem. Auto oxidation is another term sorry, oxidation in which the oxygen present is in the air is involved, right. So, auto oxidation means oxygen present in the air itself and is affecting the drug formulation. This process proceeds solely under the influence of atmospheric oxygen. Example, oil, fat and unsaturated compound can undergo auto oxidation. You sometimes see that the preparation over the period of time may get degraded due to this factor auto oxidation, the compounds which are susceptible for oxidation. Free radicals produced during initial reaction are highly reactive and further catalyze the reaction, produced additional free radicals and causing a chain reaction. So, generally free radical produced during the initial reaction which are highly effective and reactive and further catalyze the reaction and increase the oxidation effects and may degrade the preparation. After oxidation, carboxylation is also a reaction which occurs during manufacturing and storage time. Addition of carbon dioxide into a drug compound is known as carboxylation. There is a formation of carboxylic acid group like COOH acid group. So, preventive measures is like keep the drug in airtight container so that it is not exposed to carbon dioxide. So, this is the only thing that the airtight container should be used for several type of preparations which may get affected due to air, due to oxygen and carbon dioxide. Decarboxylation means that the removal of COOH group from the preparation or drug. So, elimination of carbon dioxide from a compound is known as decarboxylation. Drug substance having a carboxylic acid groups are sometimes susceptible to decarboxylation, right. So, for example, 4-amino-salicylic acid is a good example. Preventive measures like carbon dioxide gas is passed into the solution for a minute, right. So, generally to avoid decarboxylation, so carbon dioxide gas is passed in the formulation to maintain its appropriate quantity. Isomerization It is a process by which one molecule is transformed into another molecule, isomers, making isomers, which has exactly the same atoms, but atoms are rearranged. So, different structure using the same type of functional moieties, right. ABC isomer is BAC, conversion of active drug into less active form. So, generally when isomerization takes place, so we can say that drug is converted from its active form into an inactive form. So, when it may be vice versa, yes, of course. So, the structural formula, but different stereochemical configuration may affect the chemical nature of a drug product in the formulation, and it may become a less effective type of active pharmaceutical ingredient if isomerization takes place. So, preventive measures are the product should be protected from light and heat optimum. So, generally the isomerization occurs due to the excessive light exposure and heat exposure. So, optimum pH and optimum the pH change. So, pH change has to be maintained for its maximum activity, and to control or monitor the pH of the preparation, we usually add buffers. So, types of isomerization: optical isomerization, geometrical isomerization. First, we will talk about optical isomerization. A change in the optical activity of a drug may result as a change in its biological activity. It is further divided into racemization or epimerization. So, basically a change in optical activity of a drug may result as a change in its biological activity. It is further divided into racemization, epimerization, right. So, racemization, what it is meant, it involves the optically active form of a drug into its enantiomorph. For example, by the action of heat, hyoscyamine is readily converted to atropine, which is the racemic mixture of plus and minus hyoscyamine, right. So, this is basically optically active drug into its enantiomorph, means both the forms will be available, and which it is a racemic mixture. So, the active form will be substituted with inactive form. Epimerization, it occurs with the compound having more than one asymmetric carbon atom in the molecule. Example, under prolonged storage, solution containing argometrine is decomposed by hydrolysis and isomerized to argometronine. So, this is the common other example of epimerization, that the drug argometrine will get hydrolyzed and decomposed to argometronine, which is inactive form of argometrine. Geometric isomerization. So, loss of activity due to difference in the potency exhibited by CIS and trans isomer of some organic compounds. Example, active form of vitamin A molecule has all trans configuration. Trans and cis type of configuration change or conversion from trans to cis is the example, comes under geometric isomerization. So, vitamin A molecules generally has all trans configuration, but in an aqueous solution, a component of multivitamin preparation in addition to oxidation of vitamin A palmitate sometimes isomerizes, and from 6 monocis and 2,6 dicis isomers both have low potency. So, from its trans form, when it is getting changed to cis form, so the potency of vitamin A will get lowered. So, due to these geometric isomerization, all over the overall potency of the formulation will get reduced. Polymerization, combination of two or more identical molecules to form much larger and more complex molecule is known as polymerization. So, combination when of two or more identical molecule to form much larger and more complex. For example, polymer basically it is a multi-branched chain of monomers. So, when monomers interconnect, so these structures are known as polymers, right. So, polymerization basically it is a combination of two or more identical molecules to form much larger and more complex molecule called as polymer. Degradation of antiseptic formulation, and aldehyde is due to polymerization. So, when this type of reaction or process occurs in the formulation while formulating or keeping in during storage, formaldehyde, so polymerization may get degrade the overall preparation. Formaldehyde solution may result into formation of white deposit when stand cold. Okay. So, degradation of antiseptic formulation and aldehyde is due to polymerization. Formaldehyde solution may result in formation of white deposit when stand cold. So, these are some incompatibilities observed due to polymerization. Preventive measures may be: manufacturer should supply the product as a dry sterile powder. The instruction should be to dissolve the drug before use in carbon dioxide free sterile water for injection. So, these are the preventive measures that the dry sterile powder, for example, when it is to be dissolved in carbon dioxide free sterile water for injections. If the carbon dioxide is there, so it may promote the polymerization process and the overall potency of the drug will get reduced. Microbiological degradation. So, contamination of a product may sometimes cause a lot of damage and sometimes may not be anything at all. So, sometimes the microbial contamination, in majority of cases microbial contamination may affect the formulation very badly, and thus it is dependent on the type of microbes. So, level of toxicity it may produce. So, microbial presence in liquid preparation mainly may get may results in the toxicological effect, or kind of degradation, decomposition of the drug product into another deactive form, inactive form. So, basically microbial contamination and degradation is of prime importance in terms of pharmaceutical, ophthalmic preparations and parenterals. Right? If they are contaminated, they may cause serious effects. So, if parenterals or ophthalmic formulations are contaminated due to microbes, it may cause serious harm to the body, because they are the parenterals are directly reaching to systemic circulation. We are administering directly to the blood circulation, and if microbial contamination is there, they may grow in the blood circulation and cause the severe effects, side effects. So, microbial degradation, pyrogen, which are the metabolic products of bacterial growth, are usually lipopolysaccharides, and they represent a particularly hazardous product released by gram negative bacteria like E. coli, Escherichia coli. Right. So, basically pyrogens are the metabolic products of bacterial growth, or we can say that gram negative bacteria, and they may be hazardous to the preparation if present in more specifically we talk about parenterals and ophthalmic preparations. If administered inadvertently to a patient, they may cause chills and fevers, and ultimately in some cases they may cause death as well. So, preventive measures like microbial degradation, suitably design the containers. Yes, of course, when we prepare parenterals, so we say that or water for injection and sterile water for injection can be used for making these particular type of preparations. But sometimes when we put these preparation into a container, so the container if not up to the mark may contaminate the whole preparation, and it will like transferred from the container to the patient blood. So, it will grow and cause the chills and fevers and may harm the patient. So, apart from that manufacturing concern or formulation fundamental concerns, we must check the quality of the container into which we are going to keep our preparation. So, because the preparation is in directly contact with the container, and if the container is not suitable for the preparation, it may cause the contamination of the preparation. Usually single dose containers can be preferred to avoid microbial contamination, because once it is opened specifically parenteral preparations, once it gets open, so it was directly in the contact of outer environment. So, microbes or microbes are present in the outer environment, and it may affect may enter to the preparation, and once we use the preparation for single time, when we keep it for next time purpose. So, we may see that some turbidity and some sort of presence of microbes can be seen, and which leads to microbial degradation, and it cannot be used further. So, the presence of microbes can be seen using like the transparent preparation will get turbid or opaque. So, these refers the microbial growth or presence of microbes within the formulation. Sticking to proper storage condition. So, storage condition must be followed properly to avoid contamination of the preparation, or adding an antimicrobial substance such as preservatives. So, preservatives are important part of the liquid preparations basically to avoid microbial contamination, right. So, basically this is like microbial contamination is of important concern when we are administering drug directly into the blood. Sometimes like syrup and suspension and elixirs, preservative needs to be added, although it is preferable to not use preservatives in the formulation, but we cannot accommodate the presence of microbes in the formulation. So, we have to add the preservative in limited quantities. So, these are comes under the excipients category. So, method for detecting chemical and physical degradation. What are the different methods? Although other analytical methods are available, but these are some of them, like thermal analysis. Following methods can be used for detection: differential scanning colorimetry, differential thermal analysis DTA, differential thermogravimetry DTG. So, these are method for detecting chemical physical degradation. So, apart from this, there are lot of analytical techniques which can be used to identifying the incompatibilities between the drug and the excipients we use. So, any kind of physical or chemical degradation or instability occurring in the formulation, we can have the pure form of drug, and we can measure these particular parameters using these techniques, and when we make formulation, so we can check the formulation stability using this. So, we can have a better idea, like for pure drug, if it is a particular value and the formulation if it is giving the negative value, this is the data change on both the value for a same technique. So, this means the formulation is not basically stable, and it is giving the variable results. So, sometimes it is like affect the formulation. So, using these techniques, you may be able to know the different kind of incompatibilities occurring formulation with reference to that particular pure drug. So, once again we will take the highlights what we have covered in this particular presentation. So, in this we focused on chemical and microbiological degradation. So, chemical degradation basically changes a drug chemically in between the manufacturing process and storage. So, types of chemical degradation like hydrolysis, oxidation, carboxylation, decarboxylation, isomerization, polymerization. So, these are different chemical degradations. So, the hydrolysis, the reaction with water molecule. So, some drugs may be affected by hydrolysis. For example, for this cases, the buffers and complexation removal of water can be are the preventive measures which needs to be adopted. For example, hydrolysis are two types: ester hydrolysis, amide hydrolysis. Types of hydrolysis for the ionic and molecular hydrolysis. So, oxidation basically ascorbic acid, clove oil, vitamin A. So, changes or adapting addition of oxygen or removal of hydrogen may generally occurs during the manufacturing process, and drugs may get degraded. So, in that case, for to avoid oxidation, antioxidants and chelating agents are generally added. Auto oxidation, free radicals produced, and carboxylation, addition of carbon dioxide, and decarboxylation, elimination of carbon dioxide. So, isomerization, the same structural formula, but different stereochemical configuration like ABC to BAC. So, isomers form, and these are further divided into optical and geometrical isomerization. Optical isomerization further divided into racemization, epimerization, and geometrical isomerization basically refers to conversion of trans to cis form or cis to trans form to give the less effective drug. Polymerization, basically different monomers to join multiple units to form a polymers. So, sometimes it may degrade the quality of a drug product. So, microbiological degradation, particularly we talk about parenterals and ophthalmic, it is of a important concern. So, pyrogens should not be there, should not be present in the any kind of liquid preparations. It may cause severe side effects. So, preventive measures are to be taken, and these are the detecting techniques through which we can analyze the incompatibilities between drug and excipients in the formulations. So, these are references. I hope you understand the basic chemical degradation patterns and types of reaction occur during chemical degradation, and how they are important to understand which is specific to drug and its stability, and the drug overall when we add it into a formulation and we convert into formulation may get affected. So, for stability point of view, we need to understand the chemical and physical and microbiological factors affecting the drug stability. So, we will be continuing this topic in our next session. Till then, take care and stay safe. Bye.